Okay, in this video I want to talk about genetic modification. So when you hear the word genetic engineering, you straight away think of something involving tools, perhaps, to literally force DNA apart. That's sort of true, but here's the real definition of genetic engineering. There's a word you should familiarize yourself. Basically uses gene technology to manipulate the DNA within an organism. Okay, so remember, in the nucleus of cells, here's the nucleus, you've got chromosomes, and those chromosomes, if you zoom in, okay, are made of DNA. Okay, and genes are just sections of DNA, right? So taking apart DNA using gene technology, so taking apart the genes, is what's called genetic engineering. Okay, now in genetically modified organisms, the genetic information changes by inserting new genes. Okay, so in genetically modified, okay, it's usually genetically modified is usually written as capital G, capital M. Okay, quick way of writing it. In genetically modified organisms, okay, the genetic info is changed by inserting, okay, is a very important word, inserting new genes. Okay, modifying the genes, okay, is done by inserting new genes. Now these new genes are then copied to all the daughter cells when the parent cell divides by meiosis, okay, in humans and animals, for example. Okay, so new genes then copied to all daughter cells. I write capital D for daughter cells, okay? For all daughter cells, when parent cells, P cells, divides by meiosis. Okay, meiosis, remember, is cell division with the gametes, the sex cells. Okay, and then these modified cells will then mature. Okay, so these the daughter cells will mature, will grow into a completely new strain, strain or type of organism. Okay, so these cells will mature, okay, grow up, in other words, into completely new strain. Okay, strain is type. Okay, in other words, type of organism. Okay, for example, it could be a new plant. Let's a new plant, say, or a new type of animal. So that's genetic engineering. Let me now talk about gene technology. It's another word you should familiarize yourself with. Basically, it allows us to isolate a gene, okay, to alter it, to copy it, and to reinsert it, okay? So it allows us to isolate, okay, change, copy, and reinsert gene, okay, into another organism, okay, or into a new position on the DNA, okay? That's gene technology, okay? Now, the use of gene technology, has helped us develop larger harvests. Okay, so plants have been able to be mass produced. Plants with greater disease resistance, even fruit and veg that last longer and taste better. So that's some of the pros of gene technology. So the pros helped us develop larger harvests. Okay, larger harvests means more produce. Okay, in greater quantities. Plants with greater disease resistance. Okay, and fruit and veg that lasts longer and tastes better. Okay, here's an example. Genetically modified GM cotton. Okay, it basically contains an inserted gene. Okay, remember GM is by the process of GM genetic modification is it involves inserting genes, okay? So an inserted gene, okay, it produces a protein. So the gene produces protein that kills a special type of caterpillar. Okay, when it eats, so when the caterpillar eats the cotton leaf, it dies. Okay, and the inserted gene comes from a naturally occurring bacterium. So it comes from naturally occurring bacterium. Remember, bacterium with an M at the end. So is the singular for bacteria. Bacteria is a plural. Okay. Okay, it's called Bt cotton. That's the bacterium. So that's an example of using GN in nature. I now want to talk about biotechnology. Okay. Biotechnology. I'm sure you've heard of this term. It's another word to know. Basically uses organisms or parts of organisms in processes to make or change products. Okay, to make products better. So uses organisms / parts of organisms in processes to make or change products. Okay, example, IVF. I'll talk about that later on as well. IVF, in vitro fertilization. Basically, you're fertilizing eggs, so the female eggs outside of the human body, and then placing the developing embryo back into the woman's uterus or the womb. Okay, so IVF, that's an example of using biotechnology. Now I'm going to talk about use in agriculture. So using biotechnology in agriculture. Okay, here are some examples. Canola and rice. Now, canola is basically a crop that produces edible oil, as you know, canola oil, edible oil, and that's canola oil. Okay, now in the year 2010, WA, Victoria, and New South Wales basically allowed farmers to grow genetically modified canola. Okay, so they'll pass the legislation through, they can grow genetically modified canola. Now, how's it genetically modified? Well, it's basically resistant to herbicides. Resistant to herbicides. What are herbicides? They're used to basically to draw away insects for plants, right? So they can grow properly. Okay, resistant to herbicides that are used to control weed. Used to control weed around the crops. You know, as you know, weeds are really pesky little things that aren't favourable to farmers. And so therefore the canola becomes unaffected because it's genetically modified. And so the production costs, so the harvests are reduced because there's less canola dying or being affected by the weeds, right? That's canola. Let me now talk about rice. Now, people have genetically modified rice, white rice, into golden. It's called golden rice too. Okay, basically they use genes from daffodils. Daffodils are flowers. So they use genes from daffodils, corn, and bacteria to genetically modify it. Now the rice color changes from white to gold. Okay, due to a compound called beta-carotene. Now beta-carotene is what gives carrots their orange color. It makes them orange, and the body then converts this beta-carotene into vitamin A. Okay, so it's also healthier for us. It allows people in, say, developing countries to get more vitamin A because there's a lot of vitamin deficiencies in developing countries, even in developed countries, believe it or not. And so this is a benefit of genetic modification of food. So that's in agriculture using biotechnology. Let me now talk about uses in medicine. Okay. Use in medicine. Okay, I want to talk about gene splicing. Okay. Now, bacteria have DNA, right? They've got DNA in their chromosomes, but they also have some separate rings of DNA called plasmids. Now, let me write that down. Bacteria, okay, they have DNA in chromosomes, and they've got separate rings of DNA called plasmids. Think of plasma, right? Plasma is in a suspension of something. So if I draw, say, chromosomes, the X's, right? Let's say that's a piece of bacteria, a bacterium. Almost got it there. Okay, they've got DNA. So if I zoom in somehow, if I had a super powerful microscope and I can zoom in to a chromosome, there's DNA, right? And there's also separate rings hovering around in the chromosomes. Okay. Now, using enzymes, scientists have been able to cut these plasmids open. Okay. So using enzymes are chemicals that cause reactions, right? Scientists cut these plasmids, these rings, open. Okay, using enzymes, and they spliced. Splice means insert. Inserted desirable genes into them, into the plasmids. Okay, and I'll show you how this is done. Okay. So let's say we've got a nucleus, right? We've got DNA, sorry, the chromosomes. No, my mistake. Sorry. Let's say it's not what a chromosome looks like, but I'm zooming in, say, into the nucleus. So here's the nucleus. Inside the nucleus is chromosomes. I'm just focusing on one chromosome. So this is a big chromosome here. It's not big, I've just magnified it. Now you've got DNA. Right, the chromosome is made of DNA. Also plasmids, these rings. Now, the plasmids become cut, right? So they look something like this when they cut, right? Think of them as hoops of cotton or something, right? They cut them open. Then the DNA is removed from a human cell, okay? DNA is removed from a human cell. And then it's cut so then DNA is cut using enzyme, as I mentioned, right? To isolate a gene. And then a human gene is inserted into the plasmid, right, to form a recombinant DNA, right? So let's say that's a plasmid, and you've got DNA here. The process. So DNA is removed, and then it's cut using an enzyme, and then a human gene is inserted into the plasmid, okay, from the DNA. Continuing on, so that recombinant DNA, recombined DNA, so this recombinant DNA, okay, is put into a bacterium, right? It's put into another bacterium here. Okay, so let's say there's a plasmids in the bacterium. This is the recombinant DNA. Okay, the bacterial cells then grow and divide, okay, to produce many copies. So divide plus grow, and here's what you get. Okay, you get the recombinants all over the place in the chromosomes. Now the technology of combining DNA, so technology of combining DNA from different genes, okay, this is called, got a name, recombinant DNA technology. That's a word you should know as well. Recombinant DNA technology. Okay, combining DNA from different genes. Now, why is this a great thing to have? Because it can help people with diabetes, okay, by generating more insulin, for example, right? So good because can help people with diabetes. Okay, making more insulin. Right, insulin is used to combat diabetes, right? Blood sugar. Basically, nearly all insulin used by diabetics in Australia is now made by this method, recombinant DNA technology. Okay, all of the insulin almost in Australia used by diabetics is made by this process. So what are the advantages of gene splicing? Well, let me just sum it up. Pros of gene splicing. Okay, remember combining, that's what it means. Talked about crops. Okay, so crops resistant to herbicides. Okay, can provide more crops, in other words, more harvest. It's a better way of putting it. Number two, human insulin can be used to make human insulin for diabetics. Number three, making plants resistant to disease. So there are three good reasons of why we like to use gene splicing.